Hello, my name is Mr. Fisher, and this week our asynchronous video will be covering a special type of cell division known as meiosis, while also revisiting the cell cycle, mitosis, and disruptions to the cell cycle that result in cancer. This aligns with our biology TEAC B5C and our all-in assessment this week. So let's sit back and begin. Reproduction and growth are possible in both animals and plants because of cell division. This is the process by which new cells are created. Eukaryotes, or organisms that have a nucleus, use two types of cell division during their life cycles. Mitosis, the process by which the nucleus of the cell divides into identical daughter cells, and meiosis, the process of cell division that creates genetically different reproductive cells, also known as gametes. These are eggs in females and sperm in males. Mitosis takes up only about 10% of the cell cycle. During the steps of prophase, metaphase, and anaphase, and even telophase, replicated chromosomes are equally distributed into new cells. Mutations in genes that create proteins essential to the cell cycle control and lead to cancer. In cell division, this happens too very quickly, and the result is too many cells. If cells continue to divide uncontrolled, this condition is called cancer. Different types of cancer have different causes, but most result in changes in proteins involving the starting and stopping of the cell cycle. Some of the proteins that help start the cell cycle are called oncogenes. Proteins that stop the cell cycles are called tumor suppressors. Cancer normally results from a combination of both hyperactive oncogenes and underactive tumor suppressors. A great analogy is a car speeding down the highway. On the highway, there are no stop signs, as opposed to driving on a regular road with stop signs and signals that control the traffic. In contrast, a form of cell division known as meiosis results in genetically different cells, thanks to a process known as crossing over and independent assortment. In this process, replicated chromosomes line up across their homologous pair, or chromosomes in similar size and genetic content. Meiosis consists of two series of prophase, metaphase, anaphase, and telophase, steps that result in four genetically different daughter cells that contain half the number of chromosomes from the original cell. Meiosis is a type of cell division that produces gametes. The term gametes is just a scientific way of saying sperm and eggs. During meiosis, one parent cell divides into two cells and then divides again, making a total of four cells. During all of this division, cells get shuffled around and the number of chromosomes get cut in half. So the result of meiosis is four genetically unique cells, or gametes, with half the number of chromosomes as the parent cell. Now that we kind of have an overview, let's take a closer look at how this works. Before we go too far, I want to point out once again, meiosis is different from mitosis. In mitosis, we see body cells divide and form identical cells. For example, skin cells make more skin cells, and hair cells make more hair cells. And it is a process that we use for repairing and replacing damaging cells. Whereas meiosis is all about reproduction. After all, it is meiosis that gives us the sperm and the egg. And here's a little song that may be helpful in remembering the difference between mitosis and meiosis.
Mitosis is dividing, body cells are dividing. Each chromosome of DNA copies itself. Meiosis is dividing, sex cells are dividing, so gametes can connect and mix their DNA. Twisted ladder and double helix, your DNA's a barcode for you. Humans have 23 pairs of genes and sections that code your blueprint. 1, 2, your cells are dividing for growth and repairing. 3, 4, cells during meiosis, genetically different. Mitosis is dividing, body cells are dividing. Each chromosome of DNA copies itself. Meiosis is dividing, sex cells are dividing, so gametes can connect and mix their DNA. Haploid contains one set and diploid contains contains 2 sets of chromosomes from the process. Mitosis is dividing, body cells are dividing. Each chromosome of DNA copies itself. Cell cycle is made of 4 steps. Cell grows and doubles organelles. The chromosomes moving up in the middle and double away, split into 2 identical daughter cells, just like the parent cell. Like clones, they are the exact same. Mitosis is the name. Mitosis is dividing, body cells are dividing. Each chromosome of DNA copies itself. Meiosis is dividing, sex cells are dividing, so gametes can connect and mix their DNA. Haploid contains 1 set and diploid contains 2 sets of chromosomes from the process. Mitosis is dividing, body cells are dividing. Each chromosome of DNA copies itself. Gametes are made by meiosis. Copies of code are made and swapped. To spice things up, they're split in 4. One identical with only half the code. Mitosis is dividing, body cells are dividing. Each chromosome of DNA copies itself. Meiosis is dividing, sex cells are dividing, so gametes can connect and mix their DNA.
Okay, now that we have that straight, we need to understand that the sperm and egg are different than other cells in your body. They are different because not only do they have half the number of chromosomes or genetic material, they are also specialized for the process of reproduction. A basic human cell contains 46 chromosomes, but a gamete only contains 23 chromosomes. That might seem strange, but it works out really nice when you think about an egg and a sperm when they come together, which is the process we call fertilization. When fertilization occurs, each gamete contributes 23 chromosomes. Add 23 and 23 together, and you get 46. In other words, you create the first full body cell, as we say, for us as a new human. As you know, music is also a very important part of this class. Here's another song to help us remember meiosis.
There are cells called gametes, and everybody needs to make a lot of them if they want to breed. Remember the number of chromosomes in you and me. In meiosis is half from 46 to 23. Before meiosis gets underway, the original cell must replicate its DNA. Remember the number of chromosomes in you and me. In meiosis is half from 46 to 23. Homologous chromosomes exchange information, which helps create diversity in each new generation. A reductive process of division is employed to create 2 daughter cells that are haploid. Remember the number of chromosomes in you and me. In meiosis is half from 46 to 23. Each of these daughter cells divides once more which makes a set of haploid cells that equals 4. Remember the number of chromosomes in you and me. In meiosis it's hop from 46 to 23. Remember the number of chromosomes in you and me. In meiosis it's hop from 46 to 23.
Meiosis is also a big event in the cell's life, but I should also point out that most of the cell's life is spent in an interphase. This is a phase of the cell cycle in which the cell is growing, replicating DNA, and carrying out general activities. It is during interphase that a cell gets ready for meiosis. When we learn the steps of meiosis, we can use a mnemonic device that we started to learn as well in mitosis, which is called PMAT. This stands for prophase, metaphase, anaphase, and telophase. This is helpful as these processes can get a little tricky. You can see in meiosis, we need to go through these steps twice. The first trip through the steps to create new cells with equal numbers of chromosomes. The second trip through the process of PMAT, it reduces the number of chromosomes in half. So what? That step happens through the first and second steps because we are creating cells that need to fuse together. So the first cell division, called meiosis 1, uses prophase 1. The second division, called meiosis 2, uses prophase 2, and so on and so on. That's how we tell the two parts apart, PMATs apart. Meiosis 1, as we start at the beginning, this happens before any other step. And you can remember this name because we call that pro means before, just like the beginning of a book is known as a prologue. In prophase 1, we see changes start to happen with the chromosome or the genetic material of the cell. Specifically, the chromosomes condense and meet up to form homologous pairs. Homologous is a big word, but it really just means matching. Basically, each chromosome finds another chromosome that is about the same size and structure. This matching pair of chromosomes contains the same types of genes, which allows some shuffling of the genetic information and material to take place. We call this process crossing over. It forms new hybrid or unique chromosomes. This is what eventually is going to lead to the gametes carrying very unique genetic information and why people look different, even if they're siblings. Now we are ready to move into metaphase 1, where the homologous pairs of chromosomes line up in the middle of the cell. Remember that metaphase and the middle both start with M, and it might help you recall what is happening in this step. Next, we have anaphase 1. In anaphase 1, I like to think of A as standing for away or apart, because in anaphase, the homologous pairs pull apart and move away from each other. Their goal is to reach opposite poles or ends of the cell. When the chromosomes reach the opposite poles, we call it telophase 1, and the parent cell can now become 2 cells. This is where we begin meiosis 2. At this point, meiosis 1 is done, but we have to keep going. You can see that a cell reproduces by meiosis starts out with twice as much DNA as it needs. So it must go through the process of meiosis 2 to reduce that amount. What is the first step? You guessed it, prophase 2. Now we no longer have homologous pairs. There is no crossing over like we saw in prophase 1. So the prophase 2 is a little bit boring, right? We do see that chromosomes condense and make themselves more visible. So that's at least something that is similar. In metaphase 2, the chromosomes line up in the middle of the cell, just as we did before, M for metaphase, M for middle. In anaphase 2, we still use the A as they pull themselves apart. But what happens is that the chromosomes split. The chromosomes pieces are now moved apart away from each other. This splits the genetic material, so each new cell will get 1/2 of the genetic information. All that is left now is telophase 2. The chromosomes reach the opposite poles, but remember, we had 2 cells to divide. At the end of telophase 2, we get 4 unique cells that contain half of the genetic information. In other words, these are the gametes made through the process of meiosis. To wrap up the process, let's take a look at an animated video describing the process of meiosis.
Dana, hurry, we have to call up soon.
Okay, I'm duplicating as fast as I can.
Ah, there's the music, quick, call up.
Call it time. Wait to do my is right. Everything's one is almost done. Monster stop, Brooklyn's one. Patient partners do them bow. Don't see the rest over now. Help me live with your love. Promenade around the land. Oh, great partners, chromatic, crossing logic, legend is union memory breaks the near attention pocket central need. Father's cold answers done. Hold on down, it's ready, there's one. Couples moving central plate. Tempers one, so separate. Wave goodbye to reach your goal. Partner move to the other pole. Tell there's one is mighty sad. Rolling low, that's what you hate. This is just your normal state. With some of you appropriate. Rest a while and unwind to a nuclear membrane surrounds you. Now meiosis is complete. Frankly, I'm the ones that treat. Inside of that, will divide. Organics for you inside. You will find many more the same kind. If you're wondering what's in use, also you get reproduce.
What a blast from the past. I know you enjoyed that just as much as I did. As we get close to finishing our lesson today, let's review. Meiosis is a type of cell division that produces gametes, which we commonly call the sperm and egg. The steps of meiosis can be remembered using the mnemonic PMET, which stands for prophase, metaphase, anaphase, and telophase. Sometimes we add PMET-C, standing for cytokinesis. But we learned today that we have to go through the steps twice because there are 2 divisions taking place during meiosis. We call the divisions meiosis 1 and meiosis 2. Meiosis 1 starts with the prophase 1, and we see the chromosomes condense and meet up to form homologous pairs. This connection allows for crossing over, which is the shuffling of genetic information. Then the cell moves into metaphase 1, where the homologous pairs of chromosomes line up in the middle of the cell. Next, we have anaphase 1, where the homologous pairs pull themselves apart and move away from each other, followed by telophase 1, which gives us 2 cells as the chromosomes reach the opposite poles. We now have metaphase 2, which involves prophase 2, at which the chromosomes condense again, and followed by metaphase 2, which the chromosomes line up in the middle of the cell. Next comes anaphase 2, where we see the chromosome parts split apart into pieces, and they're moved apart to move away from each other. This leads to the final step, which is telophase 2. Then we have chromosomes reach the opposite poles. The result is another cytokinesis where we have 4 genetically unique cells, or 4 gametes, with half the number of chromosomes as well as the parent cells. I know this was a lot of information for today. Please take your time, rewatch the video as many times as you need. Thank you so much for your time, and I look forward to seeing you in our live sessions this week. Have a wonderful day, and I'll see you soon.